Good morning. Good morning. Hi. So I'd like to just start by thanking the organizers and the sponsors for the opportunity to speak here. It's been a great conference, and during the sessions in which we speak with people, I've crossed paths with many people who've either done research on the retina or are interested. So I think that this is a topic that doesn't always get such coverage in the media, and I'm very excited to sort of tell you about what we know, why we think it's important to study the retina, and the effects of lipids in the retina. So I'll start by speaking about those issues, why we think this is a good thing to do, and then I'll follow up by speaking about some mechanisms through which fatty acids may act in a biochemical and biophysical capacity. And from there, I'll go into human studies, speak a little bit about that, and then, again, give my commentary and perspective on what I think the next steps are, because I think it's really a fertile ground for research, and I can explain why. But, okay, so with that, so there's this basic premise that the nature of the lipids that we're studying and their metabolites have the capacity to affect processes that are implicated in the pathophysiology of macular degeneration. And I'll speak a little bit about that, but it's about trajectories. It's about how things change over time, plasticity in a system, or the ability to change, adapt, or act in a rescue capacity. Let's say I'll give some examples of that. And also I'll speak a bit about function. And so whenever I move forward in research, I try to think of this general model. And so in the context of the studies I'll present, there will be information about the relationship between intake, either being supplements or fish, and the relation with status. So that may be a biomarker in the blood, or it may be some other peripheral marker, how this may affect structure. And the beauty of the retina is that we have a transparent cornea, and we can do in vivo imaging on the retina. So it's a great system to model. And then we'll speak about how function may be affected. So there are three key points that I'd like you to take away about the nature of these lipids. So and I don't probably have to say this in this room, but the idea is that we obviously need to eat the preformed long-chain polyunsaturated fatty acids if we want to change status. We're not efficient at converting. The other issue is the resident, so these lipids are resident in the tissues and the cells and the cellular compartments that show pathology and pathophysiology in the disease I study. So DHA is an essential structural element in photoreceptors, and DHA and EPA are present both in the retinal vasculature, and the manifestations of this disease involve a pathogenic angiogenesis in some cases, so can talk about that. I put this slide up, just wanted to make it clear that the retina is about 50% lipids. Keep your eye on the blue parts of these boxes. Most of the lipids are present in phospholipid species, so they're esterified in membranes. And so this part, so if we look at the fatty acid content, this is the DHA in membranes. EPA doesn't generally, you don't find it in membranes, but about 1 in every 6 molecules in the macula of fatty acids are DHA. And then these are just other, so it's 1 in 6, 1 in 5, 1 in 6. These are just regions in the retina, and I'll speak a little bit about, what it might look like. I have a rendering, so where are the lipids found? They're found in the photoreceptor outer segments, and so I can just show you some neat pictures about. So these are the photoreceptors. They're embedded in this in this group of cells called the retinal pigment epithelium, which basically process these tips. This is the area in which the photopigment is located. This is the area in which phototransduction or sensation of light first takes place. And if you look, there are these little stacks of membranes, and those membranes are actually where the fatty acids exist. So this is where the bulk of the fatty acids in the retina are located, within the outer segments of the photoreceptors. So the next part, I'd like to speak about the capacity of these lipids to impact processes implicated in the pathophysiology of AMD. And we've spoken a little bit. There are two main topics. One is that they can bind in the nucleus and affect the production of proteins. So they can act as natural ligands to these transcription factors that basically affect protein synthesis and the volume of protein. They also act as precursors to some very potent lipid mediators, and we heard yesterday about some of the products of the cyclooxygenase P450. And these are the processes which are affected, and inflammation is a really big deal in this disease, and so cell survival. So as far as the evidence, I'll speak about the capacity of these lipids to affect cell death and angiogenesis. So these are the cardinal features that we see in this disease. So the cells degenerate. It's a neurodegenerative disease, but it's also a vasoproliferative disease in which we have this uncontrolled sprouting of the vasculature, and that can actually cause traction on the retina and bleeding and lead to sight loss. Okay, so what do we know? The questions that we have to ask: can they act as survival or rescue factors in the first place? And then can they act as anti-angiogenic factors? And so I'll start with the model cell systems. And this is not a comprehensive review. This is some work that I've been involved in. And there's much more out there, but I just figured I'd give you guys like a 10,000-foot view. Okay, so the compartments of the cell that are affected in this disease are the photoreceptors, which I showed you back in the other slide. This layer of cells called the retinal pigment epithelium, which process, and this is a vascular system. There are two vascular beds in the retina. One is known as choriocapillaris. That's the one that's affected in AMD. There's an inner retinal vasculature that exists up here, and that's what's affected in diabetic retinopathy. So we'll be speaking about this choriocapillaris. So the first point is that if we look in model photoreceptor systems, in which we actually take photoreceptors from an animal and grow them in a dish, we're able to determine that it promotes both photoreceptor survival and opsin expression. Opsin is important in phototransduction, or the ability to process light as it comes into the eye. And we can see here, this is the production of opsin in the DHA-fed cells. And this nuclear integrity assay basically tells you how many cells are living after a challenge. And you can see that there's a lot more color on this bottom part, and that indicates the ability to affect survival. Okay, so those were the photoreceptors, which are up here. Now we'll speak about the retinal pigment epithelium. There is other evidence to suggest that a metabolite of DHA known as neuroprotectin D1 also works in the capacity to save cells when they're exposed to oxidant stress. I don't have a graph on this one, but there were standard assays that were related to cell death, and this is a result that it was able to rescue. So if we look at the model system for the retinal vasculature, one of the things we can do is plate the cells, take cells from the retina that are endothelial cells within the capillaries. We can actually grow them on a petri dish. And so if we make a scratch, which is what happens here, and measure the distance that they traverse, we can actually think about whether there's a healthy response or not. Now, there's a compound which induces migration, which would be similar to what we'd see in pathology. And so we have an idea from the scratch how far these cells are able to migrate. If we add a metabolite of DHA, a hydroxylated metabolite of DHA, we find that the distance that the cells migrate is much less. And that would be something that would be sort of an anti-angiogenic process within this model system. So that's just an example of what may happen in the choroid. So This is another assay that's related to the vascular system, and so we can actually section parts of this choriocapillaris, and we can add this growth factor again, which causes the vessels to grow out of control. But if we add the DHA, you can see that there's much, or this metabolite of the DHA, or DHA. You can see that there's much less vascular growth. So again, this is something that may prevent this pathologic angiogenesis that can lead to blindness. So so we're going to move from the model cell system now into living animals. And this is a model in which the retinal vasculature is induced to produce pathogenic angiogenesis, or this florid outgrowth of vessels. And so what we have here is a retina that's mounted. The animals are fed on a DHA-rich diet. It was 2% DHA and EPA, and they were compared to an animal on a 2% arachidonate. And what we basically do is we look for vascularization in the retina, and these blank areas are actually regions in which the cells have died. But what we find is that if we feed the animals the omega-3s, the cells are actually rescued. So you want to see this whole area filled in. So this is a wild-type animal. This is an omega-6 fed animal. And this is an animal that received omega-3s before the insult. So we're going to move into a human system now, again, sectioning this choriocapillaris. And basically we can plate these and inject them with a dye. The amount of leakage that we see in this system indicates pathology. So the green fluorescein, it's a fluorophore that actually leaks from these vessels, and you don't want leaky vessels because it leads to inflammation, or it is inflammation, is something that we can use as a metric to determine efficacy. And you can see that if we feed the animals on a standard diet, this is about what we'd expect to see as regular leakage. We feed them on an omega-6 diet, we'd see something that's similar, but if we feed them omega-3s, their vessels are not as leaky. So this is just sort of an ex vivo model of cellular leakage. So, okay, so back to these ideas of the cell death and the angiogenesis. I'll speak about humans now. So the macula is an area in the eye that we use to resolve fine detail. And basically, if you think about the disease, these are the phases. We have the healthy retina here. The first phase is there are these lipid deposits that are made, and then we have a break in the membrane, and this is just what happens when there's encroachment of these vessels, there's leakage, just to give you an idea. So this is a disease of major public health significance. It's a major cause of blindness in people over 65. There are about 3.5 million people in Europe with the disease. There are about 1.5 million in the US, so it's a big deal. This is the perceptual correlate that we'd see. So on to the epi studies. What do we know about the relationship between intake and structure and status in this disease? And so you can see that since the mid-90s, there have been a large number of studies. And what I can tell you, there are international studies, there are diverse populations. Most of these studies have people, groups in the hundreds. Sometimes they're case control, sometimes they're prospective studies, but they all have at least around 400 people. And 80% of these studies show a beneficial effect of the fish intake or the omega-3 intake. So that is what really sort of put us on track. We ended up doing a clinical trial. I'll speak about that later because I see the time is winding down, but I can speak about that last. But what do we know about these lipids as an adjuvant therapy? So I talked about the fact that we have this uncontrolled vascularization of the retina. Well, there are compounds now. The standard treatment isn't to inject an eye with this anti-angiogenic compound. We were interested to know whether if we added DHA and EPA with the injection, whether the presence of this growth factor, this thing that caused all that vascular growth, I showed you that picture of the sectioned vessel, what happens to that growth factor? It's known as vascular endothelial growth factor. And what we find is this is the group of the people without omega-3s, and they were taking these by calves, and this is the number of people with the omega-3s, and you can see that the concentration of this angiogenic growth factor was reduced with people on the omega-3s and receiving the therapy. So, okay, so just a couple quick things about the genetics of AMD and how this may relate to omega-3s, because we talk a lot about gene-diet interactions, and there are a number of studies that have been done in this field that indicate that people who have certain mutations, or what I'll call a sequence variant, respond differently to omega-3s, and those responses can be related to the disease. So what do we know? So I'm just going to go back for a second. So in 2005, we discovered a complement factor, a relationship between a mutation and AMD. It's been replicated numerous times. It's about the strongest finding from a GWAS that's ever been published. It was one of the reasons why the Human Genome Project was, considered serious, because people were able to use these genome-wide association studies to actually get some meaningful results. And the p-value is 10 to the minus 200. So, there are 200 zeros in front of that first. So this is the CFH locus, and this is another locus called ARMS2 on chromosome 10. CFH is on chromosome 1. So what is it about this mutation? So if we actually look for an interaction between DHA intake and the presence of this mutation, we find that people who have certain alleles respond and others don't. So this was really interesting because it led to a reduction in the likelihood of progressing to disease, but only among people who had a certain genotype. So we have information on this major locus. We looked at the other major locus, which again is highly significant, replicated, and this is the odd ratio. There was about a 60% reduction in the likelihood of having advanced AMD if you carried this genotype and took the omega-3s. So this is something that we will certainly follow up on, but there is a gene-diet interaction for AMD with these lipids. So we talk about personalized nutrition, and that's just something I wanted to bring. I'm going to skip this slide in the interest of time. So the idea is, if we move forward with our research, I think it's very important to think about how these lipids and the enzymes, biosynthetic enzymes, the cleavage enzymes that Bill Lanz talked about 2 days ago, their receptors that I discussed as these nuclear receptors that turn on protein synthesis, how they're affected, and what we know about the genomics of this disease. So I'm currently working on those issues, but we're trying to relate them now to the uptake, the transport, the retention of these lipids, and repair, because as you may know, they're highly susceptible to oxidative stress. They have 6 double bonds, and they're very susceptible. They're in a very bioactive cell. The photoreceptor is about the most active cell in the body. So, and the idea is actually to look for the processes that are implicated in AMD, their pathogenesis or pathophysiology, and to really understand how these genetic variations can affect the AMD in this case, whether it's the degenerative form or the neovascular form. Okay, so just to let you know, on the horizon, we have some very interesting data from what's known as an ex vivo retina. So we have a retina that's outside of the body. It's a human retina. We add DHA, and we see how that changes gene expression. Now, the point is that we also know what genes are dysregulated in macular degeneration. So if we look at the intersection of those genes, okay, so it's changed by DHA, and it's changed in macular degeneration. Okay, if we find those genes, it may tell us something about the underlying biology. And so that sort of leads me to what, this session is about gaps in research, but I see it as research opportunities. So as far as the genetics go, I think what we really need to do, and there's not really much funding around for this as far as I know, but what we really need to do is take retinas and feed them with DHA, human retinas, which is all possible, adult retinas, and see which genes are changed. The samples that I have are not from adults, so I think we really have to understand through some eye donation technique which genes are changed after exposure to the omega-3s. As I said, DHA and EPA affect gene expression, but there are no existing studies, so that is a research gap. Once we have that information, I think that's something that's going to really move the field forward and tell us about the mechanisms. So I didn't talk that much about the clinical trials. There were 2 large clinical trials. I wrote the protocol for one, so I don't have much time, but I can speak afterwards about it. There were null findings from this trial, but there were a number of design issues that we discussed in our report. And as it turns out, we ended up enrolling people who enroll in clinical trials. So these people are very educated, they eat well, they do things that are, their health-related behaviors or other things. They don't smoke. So they were not an at-risk group. And I think a major research gap is that we have to target the people who have marginal nutritional status on these lipids. And I think that was a big flaw in our study. There was another study in France that was conducted. Our study had 5,000 people. It went for 5 years, but people were going out and buying DHA and taking that DHA. So we need to find a group that won't do that. We need to find a group that has marginal nutritional status. So we're going to study people at risk for compromised status, and we're going to use what we know about those genetic markers that I spoke to enroll people. Okay, so I'm out of time, but I will definitely be around at the question and answer session and afterwards. I'd just like to thank you for your attention, and I should let my colleague come. Thank you.